Last time I had discussed with you the mechanism by which HIV is capable of causing T cell depletion. There are some more mechanisms that we have to deal. I had discussed complement mediated lysis or ADCC response, which will cause the T cell depletion. We'll see what is the other mechanism that will cause the depletion of the T cell. I've told you that GP120 will be available in the serum of the HIV infected in the form of soluble antigen. And since it has a strong affinity to CD4 receptor of the TH cell, it can bind to CD4 receptors as and when the TH cells are formed. This will be happening in the initial stage of infection of the person. Now, by binding to CD4 receptor of the TH cell, it can inhibit that CD4 receptor from binding to beta 2 domain of the class 2 MHC. And you very well know that I had discussed this when T cell activation is taking place. CD4 plays an important role together with the T cell receptor. When the T cell receptor is receiving the signal by the antigen that is presented by class 2 MHC, simultaneously CD4 receptor is establishing the contact with the beta 2 domain of the class 2 MHC. And both TCR and CD4 simultaneously give signal 1 for TH cell activation. Now you imagine a situation here, as and when the TH cells are produced, they are supposed to get activated by the antigen. But on the contrary, because of the GP120 being there, which is binding to CD4 receptor, it will prevent that CD4 receptor from binding to beta 2 domain. That will make that TH cell useless. Even if it has to get activated by the antigen presentation by class 2 MHC of any HIV antigen, it will unable to get activated. And those TH cells which remain inactive, and because of the inhibition of the CD4 receptor by GP120, those TH cells become anergic. It is a term used in immunology. It may not be there in Oxford Dictionary, but it is used in immunology. Anergy, A-N-E-R-G-Y. Anergy. It is spelled as anergy. Anergy results in TH cell total inactivation. An anergic T cell will be useless for the immune response. It will undergo apoptosis. So, and you know that the TH cells are responsible for B cell activation, and humoral immune response is dependent on TH cells. When TH cells themselves are not getting activated by the antigen, and because of the major antigen that GP120 itself is responsible for the inhibition of TH cell, then there is no possibility of the humoral immune response. So this is the inactivation of TH cell may take place because of the GP120. We'll see another mechanism that is responsible for the depletion of TH cells. Okay, so here the TH cells or TC cells are very important for combating the infection, and they undergo maturation in the thymus. When they undergo maturation in the thymus, they go through two processes of selection. One, positive selection, another, negative selection. For both these selection processes, which occurs in the thymus, that is the process of T cell maturation, not only T cell receptor, CD4 receptor also plays a very important role. Remember, I had explained to you when the self antigen is presented on class 2 MHC by thymic epithelial cells, whichever the maturing T cell comes in contact with that CD4 receptor through its TCR, that will mature into CD4 T cell. Why? Because class 2 MHC has the affinity on the beta 2 domain for the CD4 receptor. And that double positive thymocyte will lose its CD8 receptor and becomes TH cells. That is the positive selection. And I had explained to you earlier about this process. Same way, CD8 receptor is responsible for the TC cells. And this process of maturation and lineage of TH cells is very important process. Without that, the TH cells will not be emerging to perform the immune response. Now we know that when the GP120 is there in abundance in the soluble form in the serum or the plasma of the patient suffering from HIV infection, it will bind to CD4 receptor of the T cells which are yet to commit themselves to the antigen. They are undergoing a process of maturation in the thymus. The GP120 can reach the thymus and bind to naive, newly formed T cells. The naive T cells, which have both CD4 and CD8 receptor on them, yet to undergo lineation into TH cells, are also going to be inhibited. The CD4 receptor, after it binds to GP120, that becomes useless and it cannot undergo positive selection to become TH cell. And when the TH cell population itself is not going to be produced in the process of maturation, then what will be the immune response? You can imagine, because only if CD8 cells are produced, it will not be sufficient to combat the infection, because those CD8 cells are also responsible. At the same time, they are dependent on the TH cell for their activation, where the TH cells will secrete the IL-2, which is an important growth factor for both TH as well as TC cells. So GP120 can prevent the T cell positive selection. It can also prevent T cell negative selection. Any TH cell which has undergone maturation process in the thymus, once if it had not come in contact with GP120, let us presume that it underwent positive selection. But the negative selection process is also very important to keep those TH cells active without committing the apoptosis. And for that reason, CD4 is very important. And there also, if CD4 has bound to GP120, then that TH cell will not be able to undergo negative selection. This will result in, once again, depletion in the population of T cells, which in the process of maturation itself, the process of maturation is inhibited by the GP120. So that will also cause the depletion of T cells. So let us see now what is the other mechanism that can also cause depletion of T cells. Some TH cells may be infected by viruses. Some may not be infected by viruses. They are still yet to be infected. But what happens is when the TH cell is infected by the virus, its membrane property is going to change. When the membrane property of the TH cell, which is infected by the virus, is going to change, it becomes highly adhesive and starts fusing with other TH cells which are not yet infected by the virus. And this fusion will result in formation of a giant TH cell. And that giant TH cell is also called as syncytium. Please note the spelling SYNCYTIUM, syncytium. Syncytium is the word used when the nucleus of the cell present in the cell in such a way that more than one nuclei may exist without compartmentalization. Means Each nucleus is of course surrounded by a nuclear membrane, but at the same time, each cell has a nucleus. Now you imagine when the cell fuses its plasma membrane with the other cell, and because of the fusion, the cell will have more than one nuclei, and those nuclei are not separated by a septum or a membrane. They are all present in group in a particular cell, and that is called as syncytium. So this is initiated by a viral protein. That virus which has infected the Th cell through the CD4 receptor now will command that cell to produce a special protein on the membrane, and that protein is responsible for causing the fusion of the other cells which are not yet infected by the HIV virus. Now let us imagine like four cells, Th cells, normal Th cells, which are not yet infected by virus, fuse with one infected cells, one infected Th cell. Then that particular syncytium will have five nuclei. Same way, it could have six nuclei, hexanucleated stage, pentanucleated stage, or tetranucleated stage, or trinucleated stage, depends on how many cells are fused with the infected cell. So that will produce a giant Th cell. Now, such a giant Th cell will become the reservoir for the virus to multiply. And that Th cell, which fused and formed a syncytium, will never be able to perform any immune function. It will never be able to produce any cytokines. It is not going to undergo proliferation. It will not show mitosis. It will be just a reservoir for the viral genome to multiply. The viral genome will multiply in that cell, and that cell becomes the reservoir for the virus to multiply and emerge from that syncytium. And millions of viruses will emerge from that syncytium, and those viruses will be responsible for reinfecting the other Th cell population which are there in the body of an individual which are not yet infected. So this is how the HIV infected person's Th cell can become useless by fusion. There are other viruses, not only HIV virus, there are other viruses which do have this ability of fusion. They cause fusion because of some special protein that they produce, which comes and occupies the membrane of the infected cell and facilitates that cell with fusion with the normal cells which are not yet been infected. And that is the tactic or the mechanism used by the virus to increase the surface area of the cytoplasm so that it can multiply in large numbers in a large surface area, that is large volume of the cytoplasm, so that it can spread the infection much faster than expected. So this is how the Th cell population can undergo depletion, that is by formation of a syncytium. Then we will see other responses. As it is, we have seen now the depletion of cell is constantly taking place in the HIV infected person, and it is an ongoing process. As and when new batch of Th cells arise, they are going to be infected by the virus. So there is continuous depletion of Th cells. Now, what will happen to the immune response, which is Th cell dependent? Now, the one of the important response that is Th cell dependent is the CTL response, that is cytotoxic T lymphocyte response. And cytotoxic T lymphocytes are responsible for identifying the cells that are infected by the virus, which is an intracellular pathogen, and cause the killing of that infected cell. Here, the CD8 T cell population has to increase in number to bring about this mechanism of cell killing. identifying each and every cell that is going to present now on the class 1 MHC, the viral antigen. And once it presents the viral antigen on class 1 MHC, we had seen how CD8 T cells are capable of causing cell lysis. That is, by getting themselves attracted to the infected cell and bind to that cell. And once they bind to that cell and establish a synapse with that cell through their TCR, which is in contact with the class 1 MHC. At the same time, CD8 is in contact with the alpha 3 domain of the class 1 MHC. And then other signals are responsible for the activation of that CD8 T cell. To some extent, the CD8 T cell itself will produce the IL-2 as an autocrine factor. To a large extent, that CD8 T cell is dependent on IL-2 secreted by the TH cell. So because the TH cell is not available now, which is being depleted by the viral population, the IL-2 will not be there in circulation. That CD8 T cell will have deficiency of the IL-2 and therefore it cannot proliferate. It will lose its ability of cell killing. It is also dependent on interferon gamma that is being secreted by the Th1 cell, which is not happening now. Because of that, the CD8 T cell will not be able to bring about cell killing of the virally infected cell. Initially, when the HIV infection has taken place, the person's disease is not at or in the advanced stage where a fresh infection has taken place, these responses will be good. They will be and they will try their level best to combat the infection, try to kill the viral infected cells, but the virus will soon overtake all these cells and the cell killing mechanism also fails. Therefore, the CTL response, when it is not there, viral proliferation will be very, very easy. And intracellular virus, that is HIV virus, will proliferate and more number of TH cells will be infected. And because of that, the viral load will be high in the individual and the immune response goes down and the person suffers from immunodeficiency. Then comes the another important response, which is Th1 cell dependent response, that is called as DTH response. Remember, I had mentioned that DTH response is very, very important to, in fact, get attracted or identify the cells that are infected by intracellular pathogens. It could be viral pathogen or it could be bacterial pathogen, it could be protozoan pathogen or it could be fungal pathogen. I've given the list of fungi, list of protozoans and list of bacteria as well as viruses which are opportunistic and they will infect the person when the immune response is very low. And you know very well, at this time, when the person's HIV infection is advancing, his TH cell response, when it is not there, and the TH cell depletion has taken place, at the same time, the TH cell dependent humoral response is also not taking place, the person becomes immunocompromised. I have used this word immunocompromised about the immunosuppressive agents. Remember that? So immunocompromised person is the one who has immunodeficiency. It is now virus that is causing immunodeficiency. And because of that immunocompromised nature of the person, all opportunistic pathogens will start infecting the person. Say, for example, the protozoan pathogens like the pneumocystis, toxoplasma, Entapheba histolytica, all these are opportunistic pathogens of protozoan origin, and they are all intracellular. Same time, the person can have infection by bacteria, which are intracellular bacteria like Haemophilus influenzae, Neisseria gonorrhea, and Neisseria meningitidis. Okay, it could be even tuberculosis. In India, tuberculosis infection is predominant in HIV patients because the tuberculosis vaccination has been done to the person. That doesn't mean that the person need not suffer from AIDS infection. That tuberculosis bacteria becomes opportunistic when the person's immune response is very, very low. And it is this time that is when the HIV infection has taken place, despite the person being vaccinated, the HIV infected person will get tuberculosis infection because at that time his memory cells, that is B lymphocytes or memory T cells, which are responsible for giving the protection to him due to acquired immune response, will also not be functioning because the desired growth factors for these cells to get activated when they come in contact with the antigen as a booster response will not happen. The fungal infections like Candida albicans and Histoplasma gondii, it is called as Histoplasma gondii, would happen in the patient. And you will see the HIV patient is a reservoir of all sorts of infection in addition to HIV infection. It is the terminal stage of the person. You cannot save the person from these infections because there is no way you can boost the immune response against these pathogens. Otherwise, naturally, these pathogens would have failed to infect the person. Why? Because his CD8 T cells would have been active, his NK cells would have been active, his TH cells would have been active, and B cells would have been active, which would have culled this infection and prevented that infection. But unfortunately, because of the depletion of TH cells, the person will suffer from multiple infections from different pathogens. Infections like Epstein-Barr virus and cytomegalovirus are opportunistic virus infect the person. So ultimately, the end result is because of these series of multi-pathogenic infection, the person will suffer a lot. The person will suffer a lot, and there is going to be multiple pathological symptoms in the person. At the same time, please remember the HIV virus is also known to cause a cancer, tumor, and that cancer or tumor is nothing but Kaposi's sarcoma. It is called as Kaposi's sarcoma. That Kaposi's sarcoma is restricted to the dermal layer of the skin, including the muscle layer that is present below the dermis. And the person will start developing nodules skin surface. It will start developing nodules, and those nodules will have black patches on them because of excessive melanin production by the melanocytes, and it is involving the melanocytes as well as the dermal layer and the muscular layer, and those Kaposi sarcoma or nodules will appear all over the body. Sarcoma refers to the muscle cancer, and these nodules will become obstructive to the person, obstructive and very, very painful, very, very painful because they will be erupting from the skin surface. And this is the last stage of the HIV infected person, where there is no possibility of saving him because no drug is going to work and no medication is going to work and no care is going to work. And this is the stage where the person's immune response is completely debilitated and compromised and is suffering from total immunodeficiency. Let us see what happens with the antigen presenting cells. Okay, antigen presenting cells, how they are made totally useless by the HIV infection. Okay, macrophages are one of the most important antigen presenting cells, and they can encounter any virus, any bacteria, and you know that these bacteria, when they infect, these macrophages can engulf these bacteria, the opportunistic pathogens that have infected. At that time, macrophages can engulf them engulf them and destroy them. But at the same time, remember, the macrophage also has a CD4 receptor. Like the Th4 cell, macrophage also has CD4 receptor, and that becomes the target of GP120. And when GP120 binds to CD4 receptor, it can help the virus that is having GP120 intact on its membrane be engulfed by the macrophage. The virus that is having GP120 intact on its membrane, that is on the spike protein, can be phagocytosed or endocytosed through the CD4 receptor in contact with the GP120, and the CD4 receptor of the macrophage becomes the facilitator for taking in the virus by the macrophage. Once the macrophage engulfs the virus, that macrophage will be unable to do the normal function. It is supposed to now fuse that endocytic vesicle or phagocytic vesicle with the lysosome. The virus that is present in the endosome or phagosome will prevent the fusion of endosome with the lysosome, and there will be no lysis of the virus. And that macrophage becomes the reservoir for the virus to multiply. And that macrophage also may develop the fusion proteins on its surface, just like the way Th cells are developed the fusion proteins, and make that macrophage a syncytium, and the virus will proliferate in the macrophage, and the macrophage becomes the reservoir of that virus. And because the macrophage becomes the reservoir, that macrophage will be now unable to do further any phagocytosis, unable to do oxygen mediated lysis, unable to do oxygen independent lysis. And it will not be able to process the antigen and present it on class 2 MHC. An inability of the macrophage to present the antigen on class 2 MHC will make that macrophage a useless antigen presenting cell. Further, phagocytic ability of the macrophage is affected. But only thing it will do is, because it has now engulfed a foreign particle, that is the antigen, that is virus, it will remain active continuously, but will not be able to do any other immune functions other than secreting the cytokines like IL-1, IL-6, and TNF alpha in excess. IL-1 secreted in excess and IL-6 secreted in excess, and TNF alpha will cause inflammatory response. It is responsible for highly inflammatory response. But what is the use? That inflammatory response will not be able to curb the infection. Th cells are not there. Phagocytic macrophages are not there. They are not able to do their normal function. Only cytokine is being produced in excess, and that will start acting on the muscles of the patient. Excessive IL-1, IL-6 and TNF alpha becomes toxic to the person, resulting in toxemia and loss of muscle mass. And that loss of muscle mass results in slimming of the person, which is called as slimming syndrome. The person shows a wasting disease. A wasting disease is nothing but loss of muscle mass. Finally, he is just skin and bone. Most of the skeletal muscles are undergoing now degeneration, and that degeneration is brought about by excessive secretion of IL-1, IL-6 and TNF alpha. And this is the terminal stage of the HIV infected person, which is brought about through the antigen presenting cells, which have become useless, have become the reservoirs of HIV infection, because they are unable to process the virus and destroy the virus, because the virus inherent ability of prevention of fusion of endocytic vesicle with the lysosome. Let us see what happens with the B lymphocyte. B lymphocyte, which have emerged fresh from the bone marrow after the maturation process, will have got the MIgM receptor on their surface. And that MIgM receptor can show low affinity to the foreign antigen and or high affinity to that foreign antigen. Whatever may be the case, on showing affinity to that foreign antigen, the B lymphocyte is supposed to undergo activation process. It is observed that the activation of B lymphocyte is TH cell dependent because they depend on the cytokines secreted by the TH cells. Now, what happens is when the TH cell population is very low and there is no TH cell population because virus has targeted the TH cell, the newly emerging B lymphocytes will establish the contact through their MIgM receptor with the soluble antigens produced by the virus, that is GP120, GP41, GP160, and P17 and P24. These are all soluble proteins which are released by the virus into the plasma or serum of the infected. And these have the affinity to MIgM receptor. By binding to MIgM receptors, even though they try to initiate the activation of the B lymphocyte, the B lymphocyte activation is not going to take place. The reason being, the TH cells are not available. B cell is not going to establish the contact with the TH cell. That synapse is very important. Synapse formation with the TH cell by the B lymphocyte is very important for TH cell dependent activation. At the same time, the TH cell secreted cytokines like IL-6, IL-4, IL-1, IL-2, all these are not available for the B lymphocyte for undergoing the differentiation process to start becoming the plasma cells. B cell growth will not take place, B cell differentiation will not take place, and GP120 interaction with the B cell is known to suppress B cell activation. And all that will result in the B cell inactivation, and inactivated B cell will undergo apoptosis. It will not be able to produce the antibodies because it cannot differentiate into plasma cells. And plasma cell differentiation process is totally TH cell dependent because the cytokines secreted by the TH cells are very important, which are not available now, and that B lymphocyte becomes useless. So humoral immune response also not there in the person, and therefore the antibodies are not there and the infection can go on. Then lastly, about the poor NK cell response. The poor NK cell response is because the NK cells are innate immune cells. Please remember that. They are innately capable of killing the virally infected cells. And they are capable of doing that. Initially, they are very good at it when the HIV infection has taken place in the person. Large number of HIV infected cells will be killed by the NK cells. And initially, HIV infected person will have the NK cell population more than 35% in his circulation. Their population will increase to such an extent, and that is one of the indicator. When NK cells are tracked and found that their population is more than 35%, one should look for whether there is intracellular pathogenic infection. And that investigation should go on thoroughly, and that is the indicator. Initially, this will go on very well because cells will be dependent on IL-2 interferon gamma secreted by the TH cells. There are some good TH cells are available which are not yet infected by the HIV virus, and they are providing this cytokine, and the NK cell is activated and it is acting on the infected cells. But as the TH cell population depletes, because the viral infection continues, because the primary target of the virus is always Th cells. And because of the Th cell depletion, complete depletion, those cytokines responsible for the further activation of NK cells will not be available. That is IL-2, interferon gamma, and IL-12, which are very important cytokines to keep the NK cell active. Proliferation of NK cells will be dependent on these cytokines, and then further action on the infected viral cells will be dependent on these cytokine activity, which is not happening. Therefore, whatever the NK cells that were there, they will have done their work, and there will be no new emergence of the NK cell population. New NK cell population will not be emerging, which is Th cell dependent. Because the cytokines are not available, no proliferation, no emergence of NK cells, and that will make very convenient for the virus to proliferate, multiply in the infected cells. So the infected cell killing may happen in the beginning, in the fresh infection, but will not happen in the later stage. An NK cell population depletes continuously, and there will be no NK cell population at the terminal stage of the disease of the person infected by HIV. And that NK cell response, which coincides with the DTH response, remember that DTH and NK cell are both responsible for getting rid of intracellular pathogens. And whenever the intracellular pathogens are there, these two responses are very, very important. And both are not available at that time. And hence the person will have to suffer from the infection continuously. And this is the terminal stage. So here I finish with the HIV infection related immunodeficiency in the person, how it causes almost all immune responses in the person depleted, including humoral immune response as well as cell-mediated immune response, and plus the responses that are responsible for the killing of the viruses. Okay, that is cell killing activity, cytotoxic activity of TC cells also depleted, the cytotoxic activity of NK cells also depleted. Initially, ADCC response will be responsible for the killing of Th cells which are infected by the virus. That is responsible for depletion of the Th cells. But later, ADCC response will not be there. When the antibody is not there, where will be the complement mediated lysis? Complement mediated lysis also will not be there. So all in total, it is a total immunodeficiency. It is a total immunocompromised condition for the person. So therefore, HIV infection is called as immunodeficiency syndrome, which is a kind of immunodisorder brought about by the virus. Therefore, it is dealt in that unit 4, immunodisorders. Okay, thank you.